Hello student, morning to one and all. I'm Dr. Tariq, Department of Biotechnology, Islamic College 1 Body. So we discussed about the topic is the subject molecular, microbial biotechnology. The topic is molecular diagnosis of infectious disease of tuberculosis. Last week we discussed the same topic in the disease of malaria. Now we discussed and elaborately described in TB, tuberculosis. The topic is molecular methods in diagnosis of tuberculosis. So actually the medical process, the progress of evaluation or revaluation, the revolution as well as the evolution, the historic drivers of medical progress. In this connection, the differential diagnosis, but that means the clinical expertise we diagnose in different aspects of diagnostic studies, in particular affected disease person. Some of the time it's genetically formation, some of the time it's naturally available. The risk assessment and the prevention also is there. It is the classical epidemiologist disease, more differentiated at the same time molecular understanding of pathological and drug action. So that molecular diagnosis is a definition of molecular diseases. Actually, the molecular diagnosis is why? Because exactly you have to identify what is the stages and status of that. That means detection and diagnosis, uncultivable or difficult to culture, need for rapid answer, inadequacy of phenotypic methods, that means biochemical aspects of studies, prognosis and management studies, its need for quantitative information, and susceptibility testing, that means drug resistance without culture, and also molecular resistant testing also is available in the diagnosis of the method. Actually, the molecular test for detection of nucleic acids in advanced stages, that means modern technology. The majority of molecular tests have been focused on detection of nucleic acid, both the DNA and RNA. There are specific to Mycobacterium tuberculosis by the techniques of amplifications such as PCR, that means polymerase chain reaction, and sometimes the detection of mutations in the genes that are associated with resistance to anti-tuberculosis drugs by the sequencing of nucleic acid hybridization. In recently, it developed in direct and rapid detection of mycobacteria. with emphasis on Mycobacterium tuberculosis species. Its identification of 16S or RNA gene sequence analysis, and at the same time oligo hybridization and strain typing also. It is as well as the detection of drug susceptibility patterns all contribute to these advances. This is called as molecular testing for detection of nucleic acid conditions. In those days, we are only for symptoms, then you have to test and blood tests, etc. Then you have to identify its formation of the tuberculosis. Very long time, the patient will come prolongation stages, end and finalist stages, because the patient are inability, awareness is very low, so that they are come and checking the finalist the disease. Nowadays the molecular studies, it will become just for sign symptoms, they are come and entirely details and molecular studies aspects, so that the beginning of gene amplification methods, the molecular studies tuberculosis, the gene amplification can achieve the goal of reducing the generation time of microorganisms to minutes, and of replacing biological growth on artificial media by enzymatic reproduction of nucleic acid in vitro conditions. The importance of nucleic acid amplification methods lies in their wide applicability in the life science and their potential to revolutionize and practice of medicine. For the example of nucleic acid sequence analysis and genetic fingerprinting, also the best methods in the amplification method. In those days, the patient tissue samples will collect and detection, and then identify the disease or formation condition. Now the new techniques, the patient tissues sample at the same time blood samples, collect the genomic studies and proteomic studies. That means using the mass spectrometry, DNA will sequencing the PCR technique also. Then it will be microimagination, proteomic imagination. Image will be displayed, so that we final and conclude the every diseases, the molecular studies aspect, it's easily detection. Actually, the molecular diagnosis of tuberculosis, rapid and sensitive tools for the diagnosis of tuberculosis that are needed due to the increased incidence of TB epidemic and the length of time required by classical diagnostic tests, especially for human immunodeficiency virus, that means HIV, it's infected persons. In this context, the recent advances in cloning and the characterization of molecular tuberculosis genes has allowed to application of basic molecular biological technique to the examination of clinical samples such as sputum and bronchoalveolar lavage BAL for the molecular diagnosis of TB infection by using the polymerase chain reaction for the amplification of microbacterial nucleic acid and non-radiometric revelation techniques. The time required for the identification of mycobacteria has been considerably shortened, 1 to 2 days, in comparison to the time required by microbiological tests. Actually, the breakthrough with molecular techniques. In this technology, the rapid development and availability for a variety of new molecular genetic technologies, the patient present the clinician with the array of options for the accurate diagnosis of infectious disease like tuberculosis. This is particularly the case for TB, since molecular methods have been rapidly introduced into all working areas of the mycobacterial laboratory, so that the Mycobacterium tuberculosis genomes were identified at the same time of analysis of the genomic aspect. We'll go through the which portion is a factor in the genomic aspect. So this is the genomic analysis of tuberculosis, particularly Microbial tuberculosis. The color is identified translation, ribosomal structure, biogenesis, transcription, DNA replication, and repair of the recombinant DNA, cell division, chromosome patienting, so cell development, biogenesis, cell motility, secretion, inorganic and transportation metabolism, signal transmission, and the energy production and conversion, carbohydrate transformation metabolism, amino acid transfer metabolism, nucleic acid coenzyme metabolism, lipid secondary metabolic biosynthesis. So many things are present in the genomic aspect. The nucleic acid amplification. In this assay, amplify the Microbacterium tuberculosis specific nucleic acid sequences using a nucleic acid probe. The sensitivity of the nucleic acid amplification in this assay currently in commercial use is at least 80% in most studies. It requires as few as bacilli of this bacteria for giving the sample. Nucleic acid amplification assays are also quite specific for Mycobacterium tuberculosis, with the specificity in the range of 98 to 99%. So that nucleic acid amplification says various types: amplificator tuberculosis assay, amplified to M tuberculosis direct assay, LC and MTB assay, and the BD probe technique, energy and transfer, and the INO lipid assay, tuberculosis assay, also the various types of the nucleic acid assay. At the same time, DNA amplification assay. The amplification of specific DNA sequences, for the example of PCR using, it provides the rapid diagnosis and the high sensitivity specification, and possible to use crude DNA, that means boiling the condition. At the same time, direct detection of Microbacterium tuberculosis in sputum by using the DNA amplification. The automatical system and some of the four kinds of methods and Roche method, PCR, 16S RNA gene abort and back to detection and gene probe, that means transcription, mediated amplification, etc. So that the manual method and single tube nested PCR for IS611 gene also, and target amplification. The target amplification PCR thermal cycle synthetic of dsDNA of hybridization of the Roche molecular system, the specific oligonucleotide to ssDNA target it extension to 65 kDa protein gene, that is called as 16S or RNA amplificator. At the same time, using the PCR 16S or RNA gene test kit also available, so that the cycle is 19 to 23. The 35 kDa protein gene sandwich hybridization methods in the assay using the Horseradis and the peroxidase, that means scabbous analysis. The analyzer is using the techniques, that means target amplification. The real-time RT-PCR is replacing older method. It is formation of these kinds of the picture shows that one. So a graphical representation is going to the high peak of this displayed in this chapter. Finally, DNA fingerprinting of Mycobacterium tuberculosis. In these studies, they very clearly and testing for one or more phenotypic markers, unusual drug susceptibility patterns, and phage typing also. These markers have been replaced by more powerful DNA typing method since the discovery of characterization of replacing DNA. The Mycobacterium tuberculosis, such as direct repeat sequence and insertion of sequences 16S or RNA, nucleic acid amplification assay. The summary is useful technology for rapid diagnosis to smear negative cases of active TB. They able to identify 50 to 60% of smear negative culture, sometimes positive cases. Distinguish Mycobacterium tuberculosis from NDM in smear positive cases only, and should not be able to use the replace sputum microscopy as an initial screen. That means, and culture remain the gold standard. Very high degree of quality control is must be required. The molecular methods of drug resistance, that is most important one, that it is the reverse hybridization, line probe assays, RNase cleavage, the diagnostic sequencing, that means genotypic analysis. In this juncture, the molecular method is more used for most using in drug resistance in the Mycobacterium tuberculosis. And finally, the molecular methods for using the drug resistance RIF ampicillin. the bind the beta subunit of RNA polymerase, and 96% of resistant Mycobacterium tuberculosis isolates have mutations in 81 base pair. This region well studied, and 4 mutations are present. 75% of resistance clinical isolated. In isonized, 2 genes are identified at the same time. These are all the 75 to 80%, and pyroxidamide in the 70%, and streptomycin is 65 to 75%. At the ethambutol and is 70% of this drug resistance in this connection. So that I conclude that each and every drugs analysis and studies is molecular studies is most important, and most technologies available in this phenomena. So that I conclude the molecular method is the best diagnosis of the particular in this disease caused TB, molecular Mycobacterium tuberculosis. So that various kinds of disease formed, and various kinds of like malaria and AIDS. At the same time, that tuberculosis is very useful and easily analysis and amplified and assays, probe, etc. It's neatly and clearly we isolated and identified, and drug resistance also formed, the good result for in the future references. So that I thanks to watching the slides. Thanks one and all. Thank you.